Hello students, today we will start lesson number 2, that is sexual reproduction in flowering plants. So in this chapter we will study the structure of these reproductive organ in flowering plants, that is the flower itself, and events or development that takes place before the fertilization, that is pre-fertilization structure and events, and after that we will study the process of pollination and fertilization. And lastly, we will study these structure and then events or developments that takes place after the fertilization, that is post-fertilization structure and events. So these are the main topics that we are going to cover in this chapter. So let us first start with the structure of reproductive organ, that is the flower itself. So this flower, it consists of 4 whorls arranged successively around this a receptacle. Now let me draw the structure of flower. Suppose this is the stalk of a plant and the swollen part, this is the receptacle. So receptacle, and this is the stalk. So around the receptacle there are 4 whorls of flower arranged one by one. So the outermost, from the outermost, if you start, we have this calyx, and next to it we have corolla. Next to it we have androecium, and after that we have, in the innermost, we have the gynoecium. So this is the calyx. Calyx is a collection of sepals that looks like a leaf, and this calyx protects the flower during the bud stage. So this androecium have this anther, bilobe anther, and then this is the stigma. Now this is the calyx. So calyx are like usually green in color. They are leaf-like structure, and then they protect this flower during the bud stage. And corolla. Corolla is a collection of petals. This is a collection of petals. This is a collection of sepals. And petals are the brightly colored part of this flower. This bright color attracts the insects for the process during this pollination. And this is androecium. Androecium means it is a collection of stamen. Okay, it is a collection of stamen, and then this stamen consists of 2 parts, that is anther and stigma. And the innermost whorl of this flower, that is this is the first whorl, second, third, and this is the innermost one, that is gynoecium. So it is a collection of carpels. We call it single. Gynoecium means it's a collection of carpels. So single piece is called carpels. So carpels consist of 3 parts, that is stigma, style, and ovary. So I'll just write it down here: stigma, style, and ovary. So this is the parts of a typical flower. And among these 4 whorls, calyx, corolla, androecium, and gynoecium, among these 4, these 2 are just accessory reproductive organs. Accessory reproductive organs. Otherwise, the third and the fourth, that is androecium and the gynoecium, are the main reproductive parts of a flower. Now we will discuss the androecium, the structure of androecium in details. Androecium means it's a collection of stamen. So stamen. We will discuss the structure of this stamen in details. So stamen looks like this. So it have a long filament, and it have a bilobe structure at the tip of this filament, that is anther. So this is a filament, and this is the anther. So anther is bilobe. So there are 2 lobes, and then it has a long, tender, slender stalk called the filament. So this is the structure of this stamen. So stamen consists of 2 parts, that is anther and filament. So this whole together, this whole structure is called stamen. This anther is a bilobe structure with each lobe having two thecas or pollen sacs or microsporangia. Sporangium, you can say. So it is called either theca, pollen sacs, or microsporangia. So these are the two lobes of this anther, and each of them have two theca. And then throughout the length of this anther, there is this longitudinal groove running. So this groove separates these two thecas. Now, if we cut this anther from this transverse section, then if we take out this transverse section of this anther, then we get a structure like this, somewhat like this. So this is the groove. This is a groove, longitudinal groove that I'm talking about here. This one, and then these are the thecas. So one lobe, other lobe. So each lobe has two thecas. So these are called theca or pollen sacs or this microsporangia. Okay. So in total we have 1, 2, 3, 4 pollen sacs or 4 thecas or 4 microsporangium. And then these two lobes, one lobe, second lobe. First lobe, second lobe, they are connected with a tissue here called the connective. So this connective connects these two lobes of anther. So this is the transverse section of this anther. Now let us study the structure of this microsporangium or pollen sac. So before also I have drawn the structure of these pollen sacs or microsporangia. Let me draw this one again. So this is the structure of this pollen microsporangium anther, the transverse section of this anther. So it's somewhat like this. So it has got 4 layers, 4 wall layers. The first one is called the epithelium, and second layer we have another second layer that is called the endothecium. And we have these middle layers here, and the innermost one, that is the tapetum. So tapetum is the cells of this tapetum is very dense. So these are the 4 wall layers of this microsporangia, tapetum. Last one, innermost one is tapetum. So out of these 4, these 3, they function as a protective structure. They are protective in function, and then tapetum, and just write down protection, and tapetum, they provide nourishment for the developing pollen grains, because pollen grains are going to be produced from this, inside this tapetum, inside this one, microsporangium. This is one microsporangium, second, third, and fourth microsporangium, or we can call it pollen sacs. So these are 4 pollen sacs, and then each of them are surrounded by 4 layers, that is epithelium, endothecium, middle layer, and tapetum. So the innermost one, they provide this nourishment for the developing these pollen grains. Pollen grains are going to be produced from here. Now the center of each microsporangium. These are microsporangiums or pollen sacs. So the center of each pollen sacs have got these compactly arranged homogeneous tissues called sporogenous tissue or microsporocytes. So in the center of this microsporangium, each microsporangium, they have got a dense homogeneous tissues called this sporogenous tissue and microsporocytes. Now this microsporangium microsporocytes, they undergo mitotic division and produce a number of pollen mother cell or microspore mother cell. So these microsporocytes, they undergo this mitosis and then they produce pollen mother cell or microspore mother cell. Later on, this pollen mother cell, they undergo another division, that is meiotic division. So meiosis, and then they will give rise to microspores. So this formation of these microspores from pollen mother cell, the formation of microspore from pollen mother cell through meiotic division is called microsporogenesis. So this is called microsporogenesis. So now when the microspores are formed, they are present in the cluster of 4. That means 4 cells, that is microspore tetrad. So it looks like this. So one cell here, another cell here, and the third and fourth cell here. So microspores, when they are formed, it looks like this. So it forms as a cluster of 4. That is why we call this one as microspore tetrad. Now as this whole enter, this is from the enter. Inside the enter there are these pollen sacs, and from these pollen sacs only these microspores are produced. So as this enter gets matured, then the pollen, this microspore tetrad, they will separate from each other. They will get separated from each other and form a single microspore. And then later on, this microspore, they will become mature and then become these pollen grains. It will be released as pollen grains. So the mature pollen grains will be looking like this. So these pollen grains will be released from here. So when the enter gets matured, from here, from this line of dehiscence, from the place where there's a long groove, this one will open, and then from here these microspores will be released. So this is how these microspores or the pollen grains are produced from this microsporangium. So several thousands of these microspores or pollen grains formed inside these pollen sacs will be released with the dehiscence. Dehiscence means the rupture of this anther. So along with the dehiscence of anther, they will come out and then it will release. So this is how these microspores are released. And in the next video, we will talk about the structure of pollen grains. So we will study the structure of pollen grains in the next video. So that's all for today. I hope you all understood.